The moment a skill becomes automatic, your brain is doing something physical. It is wrapping neurons in myelin. That is not a metaphor. It is a structural change unfolding inside your white matter, and the mechanism is not what most people picture when they think about how learning works. Something strange happens the moment you get genuinely good at a skill, not the early phase when everything feels clumsy and deliberate. The strange part arrives later, when something that once demanded your complete attention starts running on its own, and you stop thinking about it altogether. Most people put that down to repetition doing its job. Practice enough, the brain gets efficient. End of story. But the research points to something far more specific unfolding inside your white matter. And the mechanism is not what most people picture when they think about how learning works. The brain is not simply strengthening connections between neurons, it is wrapping them. Myelin is the insulating sheath that surrounds many of your neurons' axons, the long fibers that carry electrical signals from one cell to another. A type of cell called an oligodendrocyte produces it, and its function is to accelerate signal transmission through a process called saltatory conduction, where an electrical impulse jumps between exposed gaps along the fiber. Gaps known as nodes of Ranvier, rather than traveling its entire length. Myelinated fibers can carry signals up to 50 times faster than unmyelinated ones, which is not a marginal improvement. That is the difference between a dial-up connection and a fiber optic cable running the same signal through the same route. It is worth noting that oligodendrocytes are the myelin-producing cells of the central nervous system. In the peripheral nervous system, that role falls to Schwann cells, though the core insulation function is the same. For a long time, the prevailing assumption treated myelin as a developmental story. The brain lays it down through childhood and early adolescence. The scaffolding goes up, and the system is essentially fixed by adulthood. Learning, on this view, was a matter of synaptic strength, not structural insulation, and the adult brain had already completed its myelination work. That assumption turned out to be wrong. The field began to shift with a study published in 2014 led by Ian Mackenzie at University College London under Professor Bill Richardson. Working with mice, the team showed that forming new oligodendrocytes and new myelin sheaths is required for motor skill learning, not merely correlated with it. Animals that were genetically prevented from producing new oligodendrocytes ran more slowly and performed worse on a complex wheel task than controls, which meant the structure of the brain was changing not just in response to learning, but as a functional part of the process itself. Because this was an animal study, direct translation to humans requires caution, but the finding opened a serious question about what might be happening in adult human brains during skill acquisition. That question had already been framed theoretically by R. Douglas Fields, whose 2015 review in Nature Reviews Neuroscience titled A New Mechanism of Nervous System Plasticity, Activity-Dependent Myelination, laid out the case that myelin is not simply scaffolding, but an active participant in how neural circuits are tuned by experience. And here is where the picture gets more interesting. A 2016 human imaging study, carefully designed with 17 healthy adults, used a technique called multi-component relaxation (MRI) to measure myelin-related tissue properties before and after 10 sessions of visuomotor skill training involving 10,000 total movements. After training, researchers recorded increases in myelin water fraction in task-relevant brain regions, specifically the left intraparietal sulcus and left parieto-occipital sulcus. White matter had measurably changed. Related imaging approaches, including diffusion tensor imaging and fractional anisotropy measurements, have since been used to track white matter microstructure in skill learning contexts, converging on a similar story. With a sample that's small, it stands as a single study awaiting replication, but the direction of the finding carried weight. Consider also what a 2024 review of the structural mechanisms behind this process revealed about how myelin actually remodels during learning. Rather than thickening uniformly around an axon, the process appears to begin with existing myelin sheaths retracting, creating a transitional phase of intermittent myelination along the fiber. New sheaths are then added to cover the gaps, shifting the axon toward a more continuously myelinated state. The brain, in other words, partially dismantles the structure before rebuilding it in a more effective configuration, which is considerably more dramatic than anyone anticipated. The leading explanation from researchers is that neurons firing most frequently during skill acquisition become preferentially myelinated. Activity appears to signal oligodendrocyte precursor cells to differentiate and produce new sheaths around the most active circuits, so the brain is not insulating everything indiscriminately. It is selectively reinforcing the routes it is using most. Molecular markers like myelin basic protein and proteolipid protein, which researchers use to confirm and quantify new myelin formation in tissue studies, are the biological signatures of that selective reinforcement. Here is the counterintuitive part. A 2022 study from UCSF, again using a mouse model, found that new myelin formation was not required for initial skill acquisition. Animals could learn the task without it. What new myelination appeared to be critical for was the consolidation and maintenance of long-lasting memory. The structural investment arrived after the performance, not during it. Sit with that for a moment because it changes the practical picture considerably. The hours spent in early, uncomfortable practice are not the period when the brain is building durable structure. That process appears to unfold during the consolidation window, in the hours and days following practice, and research on sleep-dependent memory consolidation specifically points to deep sleep as a critical phase when newly acquired patterns are stabilized at the neural level. Compressing sessions together without adequate rest or consistently shortening sleep may not just reduce your performance. Based on what this research suggests, it could be interrupting the precise phase when structural work is being carried out. The idea that stacking more practice hours end to end is the primary lever for skill development starts to look incomplete when you look at the biology underneath it. There is something worth pausing on here that connects this mechanism to a broader phenomenon. What researchers call flow state, that quality of effortless, automatic performance where conscious effort seems to step aside, may depend on exactly this infrastructure. Circuits. Myelinated enough to operate below the conscious threshold do not need deliberate guidance. The automaticity that defines flow state is not just a psychological experience. It may be a structural one, enabled by the same insulation process described here. Honesty about what remains unsettled matters here. Human evidence in this area is still constrained by the resolution of available imaging tools, and a 2026 study published in Communications Biology illustrates both the progress and the limits. 24 healthy adults completed 4 weeks of balance training while researchers used highly sensitive multi-contrast MRI to track white matter changes across motor fiber tracts. The results pointed toward myelin-related changes in distributed motor pathways, but with a sample of that size, the findings are early, and the researchers themselves noted that the microstructural mechanisms underlying white matter plasticity during human motor learning remain poorly understood. For additional context on what happens when myelination breaks down rather than builds up, consider that in multiple sclerosis, the immune system attacks and degrades myelin sheaths across the central nervous system. The resulting signal disruption, slowed transmission, failed conduction, progressive loss of motor and cognitive function, is in many respects the negative image of what skill acquisition appears to build. That clinical reality underscores how functionally significant the structural integrity of myelin is, not just as a marker of learning, but as a foundational requirement for normal neural operation. There is also a genuine open question about what kind of practice matters. The animal and human studies that have produced myelination-related findings all involved structured, specific motor skill learning, complex wheel tasks, precise visuomotor training, balance challenges, requiring new motor coordination. Whether general physical activity drives the same adaptive myelination as deliberate skill acquisition in healthy adults is not clearly established by current evidence. General exercise may support brain health through other routes, but those are distinct claims. What the research does consistently point toward is something more specific than the usual advice about putting in hours. Skill-based practice structured around learning genuinely novel motor or cognitive patterns appears to be associated with measurable white matter changes in humans, changes that are linked to the specific circuits being trained rather than distributed broadly. And the consolidation of that structural change seems to depend on the period after practice, not practice alone. Researchers believe the brain is not simply becoming faster through repetition. The leading explanation is that it is physically rebuilding the infrastructure of the circuit, insulating the most active lines, and completing much of the structural work during the windows between sessions rather than within them. That is a different picture of learning than most people carry around, and if it continues to hold up under further investigation with larger human samples, it changes not just how long you practice, but how seriously you treat everything that happens when you are not practicing at all. The recovery window, the sleep, the spacing between sessions, none of that is passive time. Based on what the science currently suggests, that is when a meaningful part of the work gets done. If the gap between what people commonly believe about how the brain builds skill and what the neuroscience is beginning to reveal interests you, that is exactly the territory this channel covers.